Good morning. I would like also to thank Dr. Yael Ben-Chaim for organizing this meeting. We couldn't have done it without you. And I also want to thank Professor Efrat Levi-Lahad, my co-chair, who left me alone on the podium. As a hemato-oncologist, I have to admit that we are currently practicing trial and error medicine, and we are all looking forward for precision medicine. And in order to develop and implement precision medicine, we need, first of all, broad translational research of health and disease. And that's we have all over the world. But we also need big data. Having big data on a given disease will enable us, physicians, to understand what the difference or what the uniqueness of the patient in our room, and in this way, we'll be able to tailor for him the precise or the best treatment we can. In the end of the day, we need people to develop drugs and technologies, and that's also something that we have, and the more we know, the more development will be achieved. In this session, we will talk about the new ethical challenges in the age of big data and digital precision medicine. We are very lucky to have two brilliant scientists that will give their lectures, and it will be followed by debate by two brilliant other people, Professor Varda Shalev and Professor Daniel Filk. I would like now to introduce Professor Sandra Soo-Jin Lee. She is Professor of Medical Humanities and Ethics and Chief of the Division of Ethics at Columbia University. Trained as a medical anthropologist, Dr. Lee has extensive experience leading multidisciplinary research on the use of categories of race, ancestry, in genomics, precision medicine, and artificial intelligence. Dr. Lee publishes broadly in genomics, medical bioethics, and social science literatures. She currently, I don't know when you can do all this, but she currently serves as president of the Association of Bioethics Program Directors and on the U.S. Health and Human Services Secretary's Advisory Committee of the Human Pangenomic Research Consortium. Dr. Lee is a member of the U.S. National Academies of Science, Engineering, and Medicines, and she will talk on the ethics of diversity, inclusion, and global data harmonization in the age of precision. Please.
Great, thank you so much. I want to really thank the organizers of the conference and for inviting me and for the tremendous generosity and hospitality you've shown. It's been wonderful, a wonderful conference, and wonderful to see old and meet new colleagues. And I look forward to the rest of the day. Let me start by just situating myself. As you heard, I'm a medical anthropologist, and as such, my focus is really on investigating and understanding the social cultural context of biomedicine, with a particular focus on precision medicine research and its translation into clinical care. And as a social scientist, my focus on bioethical problems and their resolution begins with the point that that science is socially mediated. It is a socially mediated process in which we can think about genetics and more recently precision medicine research as a field of inquiry directed not only by the accumulation of data that produces scientific facts, but inquiry aimed at alleviating human suffering through decisions and investments that are influenced by social context, cultural values, a prioritization of specific research questions, all of which reflect fundamental understandings of the human condition. Now, I'm building on the assumptions of the work of many, but I highlight here Helen Longino, notably from her book, Science is Social Knowledge, in which she argues for the deep sociological underpinnings of science that assumes that values influence the production of scientific knowledge. And she writes, "It is, of course, nonsense to assert the value freedom of natural science. Scientific practice is governed by norms and values generated from an understanding of the goals of scientific inquiry." End quote. In this sense, she disabuses us of the notion that there is value neutrality in science, but leads us to confront, but leads us to confront how interests shape the trajectory of inquiry, what counts as an important question, what data is pursued, and how it's marshaled to answer it. Second, Longino argues that science is a collaborative process that relies on broad participation in which a diversity of values and interests can increase its capacity for innovation. So my focus today is on the problem of the gap, namely the diversity gap, as it has been defined in the context of genetics, genomics, and precision medicine. In so doing, I will draw on a current qualitative study on the conceptualization and operationalization of categories of difference that we have examined in 5 NIH-funded studies in the United States, in which researchers have responded to the call, and I think it's a ubiquitous call across the globe, for greater diversity in the recruitment of underrepresented study populations. So in so doing, I focus on the concept of diversity as embedded in what philosopher Miranda Fricker refers to as epistemic injustice. In which she argues, and I'm going to argue, that functional trouble occludes from view the underlying moral trouble that shapes the problem of diversity. So in defining what precision medicine is, it's important and helpful to understand its aspiration as a pivot away from what we've heard, a one-size-fits-all approach, to one that is described as tailored, personalized, targeted, and of course, precise. We heard from Soren the launch of the Precision Medicine Initiative in the US by President Obama in January of 2015, in which precision medicine research was a national effort in the United States aimed at a new approach to disease management that focuses on individual variability in genes, environment, and lifestyle, and importantly, relies on the recruitment of a large cohort that would provide genetic data, biological samples, and other health information to researchers. And we heard from Christine earlier about the All of Us research program in the United States that really embodies that effort. A national database of health information, including genome sequencing data that is indexed and categorized, allowing researchers to determine the genetic contribution to disease development, identify disease risk factors, and generate effective treatments that would incorporate the ways genes affect a person's response to drugs. So in short, precision medicine research can be best characterized as data hungry. It represents massive global investment to procure data from a broad swath of sources, and you can see here just the different kinds of data that is being brought in to answer this question about individual variability. And it is this effort to triangulate all of this data to identify differences between you and me and others that would presumably receive these effective diagnostics and therapeutics tailored for us. So let me talk a little bit about the diversity gap In 2009, geneticists Anna Need and David Goldstein, then at Duke University, examined the population background of samples reported in genome-wide association studies, or GWAS, and found that 96% of such studies have been conducted using samples primarily of participants of European descent. This has been noted widely in the field, and there has been growing concern about the representation in human genome research as not representing much of the globe. These findings gained wider scientific attention when fellow geneticist Carlos Bustamante and colleagues included them as part of a widely circulated call for renewed attention to diversity in genomic research. Both sets of authors expressed concern that important information about the role of genes in health would be overlooked by an overemphasis on exclusive groups or one single group. Need and Goldstein emphasized that, quote, because researchers cannot know in advance which rare variants are geographically restricted if a resource is established only in Europeans and not others, it will be more difficult to interpret the significance of rare variants in other groups, end quote. So framing the focus on diversity as a matter of benefit and equity, Carlos Bustamante went on to write, geneticists worldwide must investigate a much broader ensemble of populations, including racial and ethnic minorities. If we do not, a biased picture will emerge of which variants are important, and genomic medicine will largely benefit a privileged few. So in other words, the failure to include a much broader ensemble of populations could mean that those of non-European genetic ancestral background would fail to benefit from the highly anticipated fruits of precision medicine research, potentially exacerbating troubling population health outcomes and leading to further inequities. You can see here just some figures that try to underscore this missing data. The result of the arguments that Need and Goldstein and Bustamante and many others have made have resulted in a large number of genomic-based research initiatives in the United States underwritten by the NIH to really ask investigators to address representation in GWAS and really underscoring that there is a demonstrated bias in our data sets in terms of samples coming from primarily those of European ancestry, with the lowest percentile of genomic samples coming from those who might identify as Hispanic, Black, or Indigenous. Here on your left is Sarugo and et al.'s work in terms of the missing diversity in human genetic studies, and it puts in stark relief just how large the skew is. More recent work by Alicia Martin and her group shows us this impact of the skew in terms of polygenic risk scores and their concern about how this skew or missing data might impact the ability for precision medicine research to address ongoing health disparities between groups. So in terms of the diversity gap, there has been this direct link really focused on the threat to equity. The diversity gap has taken shape as a specific problem. It's the chasm between the portrayal of the world from the perspective of a genome data repository that is starkly different from what we know of global genetic variation. The lack of diversity as a functional problem has been named by these scientists, and in their article, George Sarugo and colleagues described the threat that the lack of diversity poses. They write, the underrepresentation of ethnically diverse populations impedes our ability to fully understand the genetic architecture of human disease and exacerbates health inequalities. Further, the lack of ethnic diversity in human genomic studies means that our ability to translate genetic research into clinical practice or public health policy may be dangerously incomplete or worse, mistaken. For example, attempts to use estimates of genetic risk from European-based studies in non-Europeans may result in inaccurate assessment of risk and lack of interventions in understudied populations. So calling upon equitable distribution of putative benefits of precision medicine research relies on a fuller, more complete understanding of the genetic architecture of human disease and the missingness of genetic materials from people of non-European ancestry threatens or stymies the potential for benefit and utility of existing genetic resources in science. So I mentioned the study that's been funded by the National Human Genome Research Institute that I lead with Janet Shim at UCSF, in which we use ethnographic methods to embed ourselves in 5 different precision medicine research studies that were responsive to the NIH call for greater diversity. And in that study, we've conducted 125 individual interviews with investigators and other research team members, as well as 450 plus hours of observation of team meetings, consortia level and scientific presentations, and then an extensive review of the research materials produced by these various teams. And from this, what I'm reporting to you today is really just one part of what we call the life course of a precision medicine research study. It's going to be focused on that part where global harmonization becomes a particular problem. But we begin by what I want to suggest is that what we find is that this issue around the diversity gap presents a dual problem. While there is much to be said about the debate about whether or not, sorry, there are different windows, whether or not and how equity should be achieved or will be achieved through precision medicine research, the focus of this talk is on the gap and how issues of datafication and harmonization can create trouble. For just as there is functional trouble stemming from missing populations, so also is there a moral problem of epistemic injustice that fails to account for missing peoples in the research enterprise. And I argue that these elisions are not merely a problem of a technical barrier solved by a focus on recruitment and retention, but a problem that occludes from view the exclusion of groups due to their unfair advantage in structuring collective social knowledge that informs genomic research and precision medicine research, and fundamental questions of what and who is studied and why. And I argue that an ethics of inclusion must address the problem of the gap by first confronting the ethical and political aspects of epistemic conduct. Okay. So to do this work, I'm relying or drawing on philosopher Miranda Fricker, who I think is very helpful in thinking about epistemic injustice and particularly the marginalization of communities that have been excluded from full participation in shaping the research enterprise. And Fricker brings into the remit of ethics questions of how actors in the research enterprise are socially situated, and builds on an assumption that social interactions and everyday epistemic practices of telling and hearing constitute social power, and that are formalized by and through legal, social, and political structures that I would argue bear directly on the problem of the gap in precision medicine research. According to Dr. Fricker, there are 2 forms of epistemic injustice, and I'm going to focus on the second one, which she refers to as hermeneutical injustice, in that there is this gap in collective resources that are shared in terms of social interpretation, where it is no accident that the disadvantage created by the diversity gap impinges unequally across social groups. And in thinking about hermeneutical injustice, as a matter of someone or some groups suffering from historically situated inequality, it is helpful to turn to legal scholarship produced by Rebecca Sosi, who is a renowned expert on federal Indian law in the United States and its impact on indigenous peoples' human rights. And she argues that the theme of discovery, which has been pivotal in scientific inquiry, has governed the violation of indigenous groups since the colonial era. And for example, she traces the trope of discovery in the 19th century in the United States as an example of hermeneutical injustice in the elision of indigenous knowledge and ethical norms about caring and sharing such knowledge. She actually describes Lewis and Clark's expedition, in which the trope of discovery, a metaphor and a rhetorical device that's been used very much in precision medicine research, was used then to lay claim to sovereign lands of indigenous nations. And the analog here is that as we think about human DNA and the search for diverse bodies and groups to address the diversity gap, thinking about the ways in which certain groups are structured in terms of their ability to produce knowledge is important. And she writes that terms such as knowledge and benefit are never neutral and are often deployed to advance or suppress particular interests and values. What we learn from Soo-Jin is that the gap cannot be understood as neutral space, one that emerges because of lack of attention, but rather a struggle over whose modes of knowing count and recognized as legitimate. So let's just turn now to the question of inclusion and how it undergirds the problem of diversity, of the diversity gap that's taking shape across the globe. And here you can see data-hungry precision medicine research, and the quest for more data has resulted, unsurprisingly, in the result of many different biobanks and genomic databases that have emerged and proliferated across the globe. This figure is from Caresses et al.'s 2021 article that provides a sense of the global effort towards data collection, curation, and the potential the potential solution to the need for large data sets. And we can see here that they're not uniformly distributed across the globe, but reflects where large public investments and centers of scientific power reside. And if we think about the challenge of harmonization, and here the challenge, of course, is how to take data from local communities in varied heterogeneous environments, different healthcare systems and systems of documentation, different EHR systems when they're available, and how do you take all of that data and make it congruent to create big data sets. This figure kind of helps us understand that process in the life course of a precision medicine research study. It requires an infrastructure that can render essentially difference into sameness. To illustrate here, this is a figure from an overview from the data harmonization process that's used by the Transomics for Precision Medicine Data Coordinating Center, or TOPMed, DCC. And you can see here an idealized process of how data is rendered and harmonized, starting with taking existing data set in these diverse formats and curated by the database of genotypes and phenotypes, the dbGaP. The data is formatted and along along with the associated metadata are stored in the DCC relational database. The critical step is step C. The harmonized phenotype variable is defined, and metadata for multiple studies are searched to identify candidate phenotype of variables. And so these various processes here are kind of a stepwise idealized approach to which you ultimately are able to render different data into harmonized data and then deposit it publicly through the GitHub repository. So there are many aspects of this process that warrant discussion and surface ethical questions. However, I'd like to focus primarily on the definitions that are used to render difference in stage A through B, C, and D, and that ultimately provides harmonized data sets to share, that will ultimately be shared with researchers across the globe. So in so doing, I'd like to highlight the challenge of incommensurability of data derived from communities with specific local histories and lived experience, and the challenge of harmonization to render basically spatial and temporal differences towards ontological completeness. And that really is the goal, to have this data set that appears to be complete and to render it congruent across different local contexts. And the question is, which frameworks, which definitions prevail on this global scale? And to illustrate some of the dilemmas, some of the barriers to doing this type of work, I want to focus on a dominant axis of diversity that is debated, is a center for a lot of debate in the United States currently, and that is racial classifications. So what you see here is, it gives you a sense of the history in the United States in terms of classifications used by the government. These are census categories. And it's had a very important role in terms of being a variable that's taken into consideration in terms of biomedical research. This has received a great deal of attention by social scientists and humanists interested in the logic of difference across groups. And it has become a vexing problem. And what you can see here is the fluidity in which race has evolved over time, defying what scientists have promoted as inherent differences between groups. We see what Howard Winant and Michael Omi, sociologists, have described as racialization, that what counts as different, what boundaries are created around groups, is socially, politically mediated, and attend to why we need to make those boundaries in the first place. And so you can see here just the ways in which different groups come in and out of view, and the different boundaries around them. And this is just to illustrate one particular axis of diversity that needs to be harmonized when you take data from different places in precision medicine research. And this gives you just another sense of the complications around those categories. This is a nice tool for teaching if you're interested in race and ethnicity in the US context, just in terms of how it's changed over a very short period of time. Now that's the US framework for difference. Let's take a look then of different classification schemes that compare some dominant systems of difference from the US to the UK Biobank to the Brazilian Initiative on Precision Medicine. And you can see here that each of these, we could talk about each of these different resources and why you see the categories that you do. They come from a particular lineage in terms of socio-political history. But I just want to highlight some of the differences. So you can see here the All of Us research program, the categories, the large categories of race are familiar in terms of the first 5, but they decided as a project to add Middle Eastern and North African. The UK Biobank here, I was trying to find myself in this biobank, and I realized that the way in which they define Asian and Asian British, I wouldn't qualify. And I'm not Chinese, so I wouldn't qualify there, but perhaps other ethnic group is where I would find myself. And then the Brazilian Initiative on Precision Medicine. And what's interesting about this particular repository is just the ways in which they combine not just race, but also geography as a way of defining populations. And just the challenges around how would you then take one sample and then decide how you're going to characterize it across these various repositories. And just to underscore the difference, here, this is what I was alluding to before in terms of finding myself. You know, where would a Korean American woman born in the United States, born in the Midwest, then spending most of my life on the West Coast and now living on the East Coast in an urban environment, how shall I identify myself, or how would I be identified using these different categories? It would be challenging. So, so this is just to underscore the what Stephen Epstein has described as categorical misalignment. Since 1993, with the National NIH Revitalization Act, researchers funded by the NIH must categorize their subjects into census categories, and as such, This has been a predominant way in which many genomic studies outside of the United States have rendered their data samples into US categories in order for the data to be collated into big data. And what you have here is one of our interviews with an investigator that really is trying to figure out what to do with this harmonization work. And he says, given his sample, just working with other studies. And so the quote here, when we first started getting studies from Brazil, I felt very silly asking them, can you please give us a breakdown? Here are the categories we've been using. They're in the US Census categories. And they responded, well, these really don't make sense for us, but I guess you could put us under Hispanic/Latino. I mean, we really don't like that, but whatever. It was interesting to try and fit people into those, I guess, wedges in the pie, and you recall Surugo's illustration of the different ancestries in the data set, wedges in the pie when it didn't feel ideal. But I guess people just want having some information. It is better than none. And so we recognized it wasn't imperfect, and yet it continued to be, their samples were labeled Hispanic/Latino. So in addressing the questions of global harmonization and data sharing, scientists, of course, and bioethics scholars have identified several challenges. Inconsistency and lack of data set interoperability due to disparate data models and terminologies. Certainly insufficient consent for data sharing that then goes out in the world after it's harmonized, and a lack of resources to support the consent process. And data privacy and security issues, as well as trying to understand the mosaic of different regulatory regimes. And while all of these are formidable barriers to broad data sharing, I would argue that these are in some ways functional trouble, and yet some of the moral trouble in terms of social justice concerns must also be addressed. So global harmonization renders data useful by making particular social stratifications between different data categories in alignment. But in so doing, the notion of the individual data subjects is often elided from view. It is inextricably bound up into populations divorced from local context. In this sense, data is dislocated and decision-making about variables of interests are redefined, and harmonization thus shapes what's counted as knowledge. So in our ethics of inclusion study, our participants recognized that barriers to meaningful translation of research findings into improved health was high stakes in terms of the kind of fragile relationships they were building in communities. And of course, these are precision medicine research teams. And citing the ways in which scientific enterprise has historically exacerbated marginalizations, some have noted that there needs to be a sense of responsibility to ensure that promises made were indeed or are indeed promises kept with regards to benefits. Research described individuals and communities as worried about the trustworthiness of precision medicine. And here one person says, you know, I don't think that science has a particularly good reputation for taking the people upon which the project design is built into consideration. And I think that many communities have very reasonable mistrust, actual mistrust of the science and the end of research at the end of research studies. Exactly like this. And I think we're asking an awful lot of people. We're asking a lot of very sensitive information. We're asking for a lot of it. And we're asking them to trust us with that. And I think if it's misused, it can both do damage to individuals, it can also do damage to communities, to groups of people, and it can do damage to that very fragile trust that communities have begun to build back with the scientific community. So I'm just going to end by saying, data justice scholars have argued that data-centric systems are instrumental as systems of control, not just in increasing potential monitoring, but as sorting mechanisms. And as such, these scholars have argued for a focus on how these sorting mechanisms work and what their relationship is to historical contexts, social structures, and agendas. Not just a question about individual privacy, but one that focuses specifically on groups and justice, and in particular, understanding the ways in which people are rendered visible, how are they represented or unrepresented or misrepresented. And I think this is a beginning point to talk about the ways in which global harmonization and precision medicine in particular takes these questions up. So thank you. And I just want to acknowledge my team, sorry. The slide disappeared. But I want to recognize my team as well as the funding that we have received through the NIH and the NHGRI. So thank you. Okay,
sure. So we have time for questions. Maybe I will start by saying that in Israel we have only 9 million people and not 33 million, but we have many diversities, and one of the experts in Israel and in the world is Efrat Levi-Lahad, my co-chair, and Efrat, maybe you want to say something about it?
For a great talk. It was very interesting. So I wanted to ask you, assuming that genetic diversity is scientifically important, what should we be doing differently? Should we be stratifying just based on the actual genetic data, so not even asking people what they perceive themselves to be, and just look at the pattern of variation? Or do you still think there is a place to ask that question of, how do you define yourself, sorry, even though I think a lot of us would have a hard time putting ourselves in any of these specific rubrics?
Yeah, no, I mean, it's a great question. So I'm not saying, I wouldn't suggest that we not characterize the samples in terms of variation. There's a very live debate, of course, that that, in terms of which axis of diversity should be a focus in terms of genetic ancestry, for example, race, ethnicity, or even genetic similarity, which is another proposal in terms of not trying to reify groups. I will say that's just one dimension of diversity, and what I'm worried, I guess my concern, if I wasn't explicit, is that precision medicine research, of course, we know, requires big data sets. And to make those big data sets and to fill that missingness that we're concerned about, there needs to be attention to what is being kind of elided from view, right? So we use these big categories, and the differences that we might think are really important, particularly for social determinants of health, which hasn't gotten as much attention, of course, but all the problems with race and ethnicity are wrapped up with all of these other variables that will then be harmonized. And what I'm concerned about is that global project, the kind of real desire to create big data sets, in that we lose a lot of the local local variables, the local lived experiences that actually might have a tremendous effect on health. And so, we use the word garbage in, garbage out. I mean, this is where certain categories will prevail. And I would actually predict that the US Census categories will prevail in terms of how samples are going to be collated and characterized. And I guess I would say I would be cautious about that because of the differences that will then fall away.
So you think there's no way to avoid the U.S. classifications? There's no way to avoid the U.S. Census classifications, even if if they're not I would say, the right ones. I'll give an example for Israel. These classifications are totally useless.
Yeah.
Because I presume I will say most Israelis would be white. There are groups like Ethiopian Jews that would probably be considered black or African, but among what we call under the category of white, you would have Ashkenazi Jews, Sephardic Jews of various ethnicities, and we would be missing out completely on diversity by using these categories. But so you still believe that that's what's going to prevail?
Wrong. I hope I'm wrong.
So, but what are we suggesting as an alternative? I guess that was my question. I mean, either we stay with the genetic determinants, which has its own problems, or we have to offer something else, right?
Yes, or, I was really struck with Christine Grady's reference to Han Jonas, this idea that perhaps slowness is not threatening to progress. And I guess I think there's a third option where global harmonization may not we might not prioritize that at this juncture because of the loss of meaning from the data and the elision of local communities and lived experience. I actually wonder if we should slow down and think about the quality of the data. I mean, this is precision medicine research, no less. And the lack of precision, I think, when you take a dominant framework of difference, like the United States in the census categories, and with a particular historical lineage, and then apply it around the world, that to me seems perilous. And that's just one dimension of diversity.